So this brings us into the concept of excitability. Right? The cell's excitability, which we'll explore a lot more when we get to neurons, but we're going to kind of touch upon it here.
Excuse me, can I ask a question? Can you go back to the last slide? Sorry, thank you. So the action potential, does it come from like the nervous system, or does it come from the endocrine system? Where is that action coming from?
That is exactly what we're going to get into.
Oh, okay,
If
sorry.
not fully
Okay, I didn't.
today, we'll
Okay.
find out where it's coming from, but we're going to really get into, you know, where the nervous system is kind of inserting into the muscle over there in the next lecture.
Okay,
we'll talk
then-
about it a little bit, but you will understand that, and you know that it's coming from the nervous system.
Okay, and then wait.
And for instance, right
I have
here,
more.
right, you see the axon of a motor neuron, right? This is a nerve cell that's going out and contacting your muscle fibers. And this is what's going to start the electrical signal, as we'll get into. But
Right.
when we talk about excitability, there's two types of cells, you know, that two types of tissue that it's excitable. The main one being, well, I shouldn't say the main one, but you know, the major one being nervous system, right? That's what it does. That's its whole function, is to be excitable and pass on signals. But the other one is muscle fiber, muscle cells, right? Muscle tissue is the other excitable tissue. And it gets excited either by the nervous system, or it has its own excitability, as we'll see with cardiac muscle, or a combination of the two, right? So, but they're all excitable. That is, they can all sort of generate some kind of electrical signal that can be passed on throughout the muscle cell and throughout the tissue in some way. Right, so let's look a little bit about what exactly that means, right? Going way back to the first unit, or was it the second one? Second unit, we talked about membrane channels, right? Remember selective permeability. The cell let in some stuff, didn't let in other stuff, and that created a difference, what was outside and what was inside, right? So there was certain types of channels, right? The selective permeability, that it was permeable to potassium, not so much to sodium. Plus you had this active transport, like pumping out sodium, right? Bringing in potassium, that was creating this electrochemical gradient, right? So this distribution of sodium ions outside versus the inside created this negative electric charge inside the cell, right? And not just that, there was a lot of, you know, your proteins, your DNA, large proteins and stuff are overall negative charge. But that extra part was that sodium was being kept out, right? Sodium would love to go in here and interact with all the negative stuff, but it was being kept out. So you have this potential, like a battery, of a positive charge relative to the negative inside charge. And the cell, this is what the cell is going to use to create this electrical signal, right? Kind of like way back when we talked about that, right? Like the potential energy of a dam, right? You store all this water, and all of a sudden you open the dam, and then all the water rushes through, and you're going to create that energy. And that's what is going to happen with this membrane. All this positive charge out here really wants to get on the inside of here, right? So, but you have this, what's called the membrane potential. The cell is negative in relative to the outside, right? You always see this number, about minus 70 millivolts, negative relative to the outside, positive on the outside, negative to the inside. That's going to be the first step. You need this to get this excitability, right? That the cytosol inside the cell, negative relative to the outside, right? So you create this sort of dam situation. Or the other way to think about it is, if this was really hot out here and this was cool in here, if you opened up a window, right, what happens, right? The hot air kind of rushes in, something like that. All right, so way back when also, we talked about different types of ion channels and how they might be gated, right? Kind of closed and open in response to something or other. All right, and so we had those leak channels that were kind of randomly open. And then we had things which in presence of a certain ligand, like something like even a hormone or something, it bounds through this membrane channel, it opens up, and then whatever was around here would flow down its or out its electrochemical gradient, right? Whatever the channel allowed through this binding of this signal over here. Another signal might be mechanical pressure, right? You press on it and then the channel opens, and then again, stuff that was kept out was going to flow in. And then the last one here is relevant to us. The last signal here is that there's a voltage change, and that voltage change opens up the channel, and then whatever the channel is permeable to will kind of flow. Those molecules will flow according to their electrochemical gradient. In this case, remember I said potassium is kept high inside the cell, right? And then the cell is normally kept at this minus 70 millivolt, right? This electric charge. If for some reason the voltage rose to minus 50, like towards zero, right? That opened up a channel, and all of a sudden potassium ions would flow out. Right? So that's what you call voltage gated potassium channel. And in that case, it's potassium, but it could also be a voltage gated sodium channel. And if you have a bunch of those voltage gated sodium channels embedded in the sarcolemma in a particular arrangement and the T tubules, you can pass on an electric signal. That is, an electric signal in the form of an action potential can propagate down the length of the sarcolemma throughout the whole cell. And it's due to the presence of these specific voltage gated ion channels. That is, there'll be a little difference here in membrane for some reason, right? It's going to be sensed by this channel. It's going to open up. Sodium is going to flow in. That's going to create a electric change, which is going to open up this channel, which is going to allow sodium, which is going to create electric change, which is going to open up this one, right? So there'll be this propagating chain of events of these channels opening up down the length of the entire cell, right? That's called the action potential versus a membrane potential, right? So here's next lecture, we'll talk about this part of it, right? What starts that action, right? But here we're just going to look at this these voltage gated. For the most part, we'll just stick to these sodium channels. The potassium stuff we'll get into when we get into the nervous system. Let's just think about the sodium ones arranged down here, creating this chain of events. This voltage flips down the length of the fiber right there. Right. So here's the other cartoon version, right? You had the membrane potential, that was just this part in yellow right here, but the action potential is this moving, right? This cascade of voltage flips that go down the entire length, right? And for muscle fiber, it'll just be going out throughout the entire length in both directions. This sort of voltage change, right? Until it reaches whatever it reaches. That's one way to generate a cascade of voltage flips is one way to think about it. Any questions on what's happening? Why we call this an excitable membrane, an ability to generate this signal going down there. And we're going to use that signal eventually for calcium release, as we'll see. So here is, this is what we'll get into when we get into the nervous system a little bit. And actually we'll get into this for this unit too. A little bit here. Here's one of those nervous system motor neurons. They come out, they have these axons, they come out and they talk to these and tell them to contract, right? So here are those muscle fibers over here packed with those myofibrils. And we'll see an electrical signal go down here. And then the electrical signal will be passed on to the muscle fibers, right? Down the entire length of the muscle cell, right? And that electrical signal, as we'll see, is what's going to cause a calcium release. So now we know about the electric. This is why the action potential is traveling down. You had all those voltage gated potassium channels traveling down the membrane, down the sarcolemma. And then because the T tubules are an extension of those sarcolemma, we're going to travel down these T tubules. The T tubules, again, we're going to be associated with the sarcoplasmic reticulum and those triads right here. And then we'll see how exactly this causes the calcium release. Right? So here's the T tubule going down here. The action potential has gone down it. And now the connection in between the T tubule and the sarcoplasmic reticulum is what's going to release the calcium. And it's going to release it all over the sarcoma here, allowing this interaction. So let's look a little bit closer at what's going on with this. Prior to the excitation, right, the action potential, you have the sarcolemma in blue right over here, the T tubules that are the little dips right over here. And then this is like a very schematic kind of picture of the sarcoplasmic reticulum, right? And inside the sarcoplasm, over here and outside over here, calcium levels are very low, right? There's very little calcium levels. And that's because you have these calcium ATP ACE pumps. That is, they're pumping calcium against its concentration gradient into the sarcoplasmic reticulum, keeping sarcoplasmic, where the sarcomeres are, levels low, right? Calcium levels low in there. They're keeping it all inside in here through this ATP ACE pump. Right? Energy is required. ATP is required so that all the calciums are kept in control inside the sarcoplasmic reticulum. All right, so this is what's happening in the before the action potential comes, right? So here's the overall state. You have the sarcoplasmic reticulum filled with calcium ions. It's kept in there by this calcium ATP ACE pump, right? So that it's filled with calcium. These are kind of quiet, right? There's no calcium in the sarcoplasmic. There's no interaction or only weak interaction really with the myosin and actin filaments. So there's no movement of the muscle. Then we get the muscle action potential right here, traveling down the sarcolemma, down those T tubules right over here, right? Within the T tubules right here, you have another voltage gated ion channel, a voltage gated calcium channel, right? As opposed to those regular voltage gated sodium channels that were propagating your action potential. These voltage gated calcium channels open up, right, in response to the action potential, and in a way the book doesn't go into, so I won't either, causes a magical, because it's interaction with this calcium release channel that's embedded within the sarcoplasmic reticulum. The interaction, the change in conformation due to the voltage change over here causes this interaction to change somehow and release the calcium out of the sarcoplasmic reticulum. Everybody clear on that? Muscle action potential comes down. Voltage gated calcium channels are within the T tubules, the membrane. Your calcium release channels are within the sarcoplasmic reticulum. That's going to release the calcium. Everybody clear on that? And if you're reading other books, you'll see there's a couple other names for these channels, but this is all you have to know. Voltage gated calcium channels, calcium release channels. Neither of these are to be confused with the calcium ATP ACE pump. Right? So we have these three types of calcium channels, all very different, right? So here we go, big picture, zoom out. Action potential comes down, voltage gated calcium channels open up. Somehow they interact with your calcium release channels of the sarcoplasmic reticulum. Calcium is released, right? Calcium spills out all over here, binds to troponin, right? Undergoes conformal change, causes tropomyosin to move away, right? Cross bridge formation happens, pivot, right? Power stroke, muscle fiber contracts, right? All right, everybody see the whole sequence of events right here. Everybody see how, if they don't get it immediately, how it's kind of understandable, right? You just want to keep all the moving parts in action right here. Notice everything we talked about here didn't have anything to do with the whole ATP of the myosin head right here. That was like a kind of a different story, right? That's happening semi-simultaneously, but you know, these are two kind of different events. Right? The ATP over here. ATP is involved where? It's also involved in this whole interaction over here, right? That's going to bring the calcium back in. All right, so that's that sequence of events, right? So we go from the excitation, action potential happens. That's going to cause a calcium release through all the mechanisms I just said. Cross bridge formation is going to occur, and then that is going to power stroke, right? Contraction is going to happen right there. Right, so that's your excitation contraction coupling. Excitation of the membrane leads to the contraction of the sarcomere. The last part of it is, again, once the calcium is released, right, it'll keep those binding sites exposed, and it'll keep the muscle contracting. That is, it'll keep the sarcomeres acting as long as calcium is there, right? As long as, you know, the ATP stuff is all, you know, doing its thing, right? So you don't want that. You want to have this strictly controlled by the nervous system input. So you want to get rid of that calcium. You want to put it back inside the sarcoplasmic reticulum, right? So the calcium ATP pump is that necessary last step to get the calcium, clear the calcium out of the sarcoplasm, so that there's not unwanted contraction, right? In fact, that's one of the things that happens if you've heard of rigor mortis. One of the things that happen is calcium, these channels break down and calcium is released, which causes that interaction, right? And since there's no ATP being formed, the myosin head doesn't detach from the actin filament, right? So your rigor mortis is when somebody dies and their muscles slowly contract, right? And they contract for like an hour or two, and eventually everything just kind of dies out. But that's why muscles stiffen when people die, right? Because you don't have this calcium ATP pump, and the myosin head doesn't detach. Okay. So for all this, you know, one of the questions in the book, you know, what are the three roles of ATP in this, right? That first one is that whole interaction where ATP is hydrolyzed to ATP to get it ready into the cocking position. And then after the power stroke, ATP is needed to release that, right? And then the last part over here is that the ATP is needed to pump the calcium back against its gradient inside to the sarcoplasmic reticulum, right? And then, of course, you know, you have the membrane potential being the ATP, but these are the ones directly related to that. Again, that's maybe a paragraph or so in your book, just these figures, but just go over them just so you understand them, and you'll be good on those. Because next time, you know, your question, the person who asked the question before, you know, where does that signal coming from? We're going to really talk about the interaction between the nervous system and the muscle at this neuromuscular junction, right? The synapse in between them, and how this happens over here.